presentation of AIT.
In our genes, directs us from behind the scenes. The words within it shape life's destiny. Hidden in your DNA is your genetic dossier. It tells your future and your history. How traits get passed from parents to a child is something that has kept us so beguiled. Cracking the code. Genetic mysteries to unfold. Cracking the code. Genetic secrets will be told. Cracking the code. Genetic mysteries to unfold. Cracking the code. Genetic secrets will be told. Cracking the code.
By the second decade of the 20th century, it was clear that within the chromosomes are stored the units of heredity, the genes. But what still wasn't clear was the physical substance of the gene. What was it made of? The first clue appeared in 1869 within an ancient castle in Germany. Inside a foul-smelling room was the lab of Johann Friedrich Miescher, an early biochemist. He was interested in the chemical makeup of white blood cells, particularly their large nuclei, and he extracted these cells from soiled bandages obtained from a nearby hospital, hence the stench. Miescher used dilute hydrochloric acid to separate out the nuclei from the rest of the pus cells. Then he extracted digestive juices from the stomach of a pig to dissolve away any remaining bits of protein. He then used various other chemicals to separate out the contents of the nuclei. What he ended up with was a strange, gummy sort of substance. Miescher tested it chemically and found it to be rich in both nitrogen and phosphorus. He called this new material nuclein. In fact, it was DNA. It was shown that nuclein is an acid, now termed a nucleic acid, which consists of a chain of smaller parts called nucleotides. A nucleotide itself consists of 3 smaller molecules joined together side by side, with a sugar in the middle, and a phosphorus-containing acid or phosphate on one side, and a nitrogen-containing base on the other. By the 1920s, it was recognized that there are 2 different nucleic acid chains. The difference lies in their central sugar molecules. One chain was named RNA, short for ribonucleic acid. The ribo refers to its particular sugar, ribose. The other was named DNA, short for deoxyribonucleic acid. Its sugar has 1 less oxygen atom than ribose, hence the prefix deoxy. It was also noticed that DNA is found mainly within the nucleus, and RNA mainly outside of it in the cytoplasm of the cell. It was also found that the nitrogen-containing bases in both DNA and RNA come in several different forms, represented by the initials of their chemical names: adenine, guanine, cytosine, thymine, and uracil. 3 of these bases are common to both DNA and RNA. Thymine is unique to DNA and uracil to RNA. At first, little notice was taken of these small differences among the nucleotides that form DNA, but these variations within their base components were ultimately revealed as the alphabet of the gene. But in addition to DNA, chromosomes also contain protein, and for the first half of the century, protein stole the show. A protein consists of an irregular sequence of smaller amino acids. There are 20 different amino acids which can be arranged into any possible combination. As the protein grows longer, the possibilities multiply astronomically. DNA, on the other hand, has an alphabet of only 4 letters, which were thought to be arranged in a monotonous sequence. It's no wonder that proteins seemed a better candidate to carry the language of heredity. It took a couple of scientific surprises for DNA to finally prevail as the stuff of the gene. The first took place in 1928 in the London lab of Frederick Griffith, a physician. He was studying 2 different strains of a bacteria called pneumococcus. One strain had an outer capsule and was highly infectious. When Griffith injected these potent bacteria into a lab mouse, it got sick and died. No surprise there. His next step was to kill these bacteria by heating them. When he injected the dead bacteria into a second mouse, nothing happened. Again, no surprise. Griffith's other strain of pneumococcus lacked the outer capsule of the first and were harmless. When he injected these bacteria into a third mouse, again as expected, they had no effect. Lastly, he combined the second strain with the killed version of the first strain. To his great surprise, this combination proved deadly. When Griffith examined the dead mouse, he found live bacteria that looked just like the infectious strain. This suggested to him that some factor had passed from the dead bacteria into the live ones to transform them. He also discovered that this transformation was maintained in all subsequent generations. So Griffith's transformation factor must have contained genetic material. Across the ocean in New York, a team of scientists at the Rockefeller Institute set out to identify Griffith's transformation factor. They were led by Canadian-born Oswald Avery, who, like Griffith, was a physician interested in the pneumococcus. Avery's team carefully separated out the chemical components in Griffith's dead bacteria and added them, one at a time, to living cultures of his benign bacteria. They found that the transformation factor was pure DNA. It was an astonishing result. After finally satisfying themselves that DNA and only DNA was the transforming substance, Avery and colleagues published their findings in 1944. Their discovery about DNA in bacteria had a major impact on a budding young scientist named Joshua Lederberg.
I was thunderstruck by it. Maybe I was at one extreme in that regard, and it changed my whole life.
Lederberg did a series of ingenious experiments to see whether bacteria can exchange genetic traits as they reproduce, like Mendel's peas and Morgan's fruit flies. In other words, to see whether bacteria have a sex life. If they do, they must have genes, even though they lack a nucleus and any visible chromosomes.
It turns out that bacteria have a very vigorous sex life. In fact, microbes, or bacteria in particular, are probably the most promiscuous of organisms, and that they can exchange genetic information over the entire kingdom, and in fact, in some ways, even across those boundaries. If we look in the human genome, there are things that are inescapably of bacterial origin in our own genome, so there was some promiscuous mating that at some earlier stage in history.
Lederberg's discovery of bacterial sex at age 20 won him a Nobel Prize 12 years later, making him one of the youngest Nobel laureates ever. Although many scientists were not yet convinced, DNA was now a serious contender as the molecule of the gene. The next challenge was to work out its structure in the hope that this would explain how a gene actually works. The solution to this challenge came through applying the non-living sciences of physics and chemistry to the living science of biology. Physics contributed through a technology developed in the early 20th century. Called X-ray crystallography, it used early devices like this to give scientists their first glimpses into the inner structure of a molecule. Almost 100 years later, X-ray crystallography is still used for the same purpose. The molecules are more complex and the equipment more sophisticated, but the basic principle remains the same. The molecule being studied is converted into a crystal, which locks its atoms into a uniform three-dimensional structure. X-rays are then beamed at the crystal. As they pass through, they are bent or diffracted by the clouds of electrons that surround these atoms. These scattered X-rays are recorded on a film or photosensitive receptor as a pattern of dots that is unique to that particular molecule.
And we can figure out from the direction of the scatter where the electrons were that produced that scattering pattern, and from that, the shape of the molecule.
By the early 1950s, X-ray diffraction of DNA revealed some tantalizing clues about its structure. But the X-ray dots were like tiny peepholes through which only certain features of the DNA molecule were visible. One feature the dots revealed is that the nucleotides in a DNA chain join together to form a backbone of alternating sugars and phosphates. The dots also showed that the DNA backbone curls around its axis to form a corkscrew or helix shape. The dots also suggested that a DNA molecule is composed of two such helical backbones winding around each other. The resulting structure is called a double helix. However, the X-ray photos were silent on the third component of the nucleotide, the 4 different bases. Because of their variability, they scattered X-rays too randomly to form dots. They therefore remained invisible. But the bases had to be within the double helix, attached at right angles to the sugars. Opposing bases must somehow bind to each other, gluing the two backbones together. It had long been known that the 4 bases come in 2 different shapes. A and G are larger 2-ring structures called purines, and C and T are smaller 1-ring structures called pyrimidines. Scientists also knew that identical bases could join together side by side through hydrogen bonding. But such base pairs are either too big or too small to match what they knew was the diameter of the double helix. Also, such base pairs could not have produced the very regular shape suggested by the X-ray photos, which resembled a smooth-walled cylinder. To fit them in, the cylinder would have to bulge out for the larger purines and shrink in for the smaller pyrimidines. The next set of clues came from chemistry. Erwin Chargaff, a biochemist at Columbia University, was also inspired by Avery's discovery about DNA. In the late 1940s, he studied DNA in various species. He found that the 4 bases were never present in equal amounts, and that each species had its own characteristic ratio of bases. But no matter which species the DNA came from, the amount of A was always equal to the amount of T, and the amount of G was always equal to the amount of C. These became known as Chargaff's rules. It was at Cambridge University in England that the various pieces of the DNA puzzle finally came together. The exact location was the Cavendish Physics Lab, which specialized in the X-ray crystallography of proteins. That is where a pair of junior scientists shared the same small workspace and the same conviction that DNA, not protein, was the molecule of the gene. James Watson was an American, a 23-year-old biologist who had recently earned his PhD mapping out the genes within a group of viruses that infect bacteria.
These viruses contain DNA, and the most plausible, given the result of Avery, was genetic material. DNA was in the chromosomes.
Francis Crick was an English physicist, 12 years older than Watson, but still working on his PhD in X-ray crystallography. Sidney Brenner is a fellow pioneer in molecular genetics who knew Watson and Crick back then. He and Crick later shared a different office near the Cavendish, where together they worked out the basics of how a gene produces a protein.
Francis was, as as one remembers him then and as one remembers him throughout the 50 years, very ebullient, I mean, very excited by everything. Jim sort of hovered around being a bit irritable.
But the two of them together were really quite a very formidable team. And you know, with hindsight, it's clear that they were the right people in the right place at the right time.
Watson and Crick would spend hours talking about DNA, often at the Eagle pub across the road from the Cavendish. They decided that together they could solve the structure of DNA, which they hoped would reveal something about its function. Their plan was to build a model, using whatever clues they could glean from X-ray data, combined with the rules of chemistry that dictate the angles and distances of the known atomic bonds within DNA. They were inspired by the famous American chemist Linus Pauling, who had recently used model building to work out the first molecular structure of a protein. In so doing, he had scooped the Cavendish lab, which was in hot pursuit of the same quarry. His structure, called the alpha helix, reinforced Watson and Crick's belief that DNA was also a helical shape. But the Cavendish only did X-ray diffraction of proteins, not DNA, which is a trickier substance to work with. But good DNA photos were close at hand at King's College London, in the X-ray lab established by physicist Maurice Wilkins, a friend of Crick's. The best photos were taken by Rosalind Franklin, a talented young crystallographer recently hired to work on DNA. Wilkins thought she was to work for him. Franklin thought, with some reason, she was to do her own research. So their relationship got off to a rocky start, from which it never recovered. There was also a definite personality conflict. Sir Aaron Klug knew both well, particularly Franklin, with whom he later worked.
He was rather shrewd, slow, systematic, careful. She was rather impulsive, fairly fierce, and so on, different personalities.
In this strained atmosphere, Franklin was in no mood to collaborate with Wilkins, let alone with Watson and Crick. Besides which, she did not agree with their model-building approach. She thought the answer would come through X-ray analysis alone, which she chose to pursue on her own.
I found in Francis, we could talk together. We really got along, and Rosalind didn't have anyone to talk to.
Watson attended a talk that Franklin gave at King's College in the fall of 1951. Most of it went over his head, and he took no notes. But based on what he thought he remembered of Franklin's X-ray data, he and Crick plunged ahead with their first model. It was a disaster, and Franklin immediately pointed out its obvious flaws. She scorned their subsequent invitation to collaborate. Their boss, Sir Lawrence Bragg, ordered Watson and Crick to leave DNA to the group at King's College and to return to the other projects they had put aside. Although forbidden to work on it, they couldn't stop thinking or talking about DNA. Then, in early 1953, word reached them that Linus Pauling had turned his formidable talents to solving the DNA structure.
Linus Pauling, of course, actually did come out with a DNA structure. A triple helix, which had a very elementary chemical flaw in it that Jim and Francis were delighted to see.
Watson and Crick took this news to their boss. They convinced Sir Lawrence that it was only a matter of weeks until Pauling got it right and again beat the English at their own game.
After Pauling came out with his proposed structure, he was concluding that people at King's weren't doing anything, and Francis Crick and I should be allowed again to see if we couldn't find the structure by model building.
Thus began one of the most celebrated races in scientific history. But only Watson and Crick knew the race was on.
Jim's book, The Double Helix, portrays this very much as a race. They were certainly worried about Pauling, there's no doubt about that. But Pauling, I don't think, knew he was in a race.
Francis said I was the only one racing. He certainly wasn't racing. Rosalind wasn't racing. Linus never raced with anyone in his life.
Watson and Crick needed Franklin's data, but she still refused to share it. In the meantime, she had made real progress. She was the first to realize that DNA produces two different X-ray photos, depending on its water content. The war-damaged courtyard of King's College was being excavated when Watson again visited in early 1953. He hoped that the news about Pauling would bring the two English groups together. After a hostile response from Franklin, Wilkins took Watson aside and showed him one of her photos of the wet or B form of DNA. Its dots formed an obvious X shape. He later wrote, "The instant I saw the picture, my mouth fell open and my pulse began to race." The X shape told him that DNA was a uniform helical structure with certain dimensions to it. He rushed back to Crick with the news. Through another bit of luck, some other data of hers fell into their hands, again without her knowledge. Together, this was enough to set them on the right track with a new model. This time they got the position of the backbones right, the basic double helix structure. But they were still stuck on how to fit the bases into it. One Saturday morning in February 1953, Watson was at his desk playing with cardboard cutouts of the four bases. He saw that identical bases could easily bond with each other, but the resulting pairs were still either too big or too small. Suddenly he realized that A could just as easily bond with T and G could bond with C, and that no other such pairings were possible. Now the resulting base pairs were the same width, and the width of the double helix matched perfectly with the width of the base pairs.
You needed that fundamental idea to make sense of things. So once you saw that, then of course it all fell into place.
We went from nothing to everything, and that Saturday morning on February 28, 1953. That was, you could say, a eureka moment.
When Crick arrived later that morning, he was greeted by a jubilant Watson. He quickly agreed that this must be the answer. Chargaff's rules now made sense. If A always paired with T, they would have to be present in equal amounts. Similarly, the amount of G would have to equal that of C. Crick also realized that the two backbones must run in opposite directions. They celebrated over lunch at the Eagle Pub, where Crick announced to everyone within earshot that they had found the secret of life. They continued with their model building, this time fitting the new base pairs into the middle of their double helix. With a bit of adjustment, they found that all the parts now fit perfectly. The two backbones were held together by the base pairs, A joined to T and C joined to G.
Adenine, thiamine, cytosine, guanine, O. Adenine, thiamine, cytosine, guanine, O. Watson and Crick pulled off a very slick trick when they unlocked the secret of the gene. The facts they knew were quite few, just the occasional clue pointing to DNA, not protein. But what was its configuration? How did it store information and allow for duplication with only 4 nucleotides? Only 4 nucleotides. How they fit together must be where the secret hides. Adenine, thiamine, cytosine, guanine, O. Then using new facts, learn when an X-ray diffracts, the pieces started to fall into places. They began to play with models that could display a way to form DNA by linking bases. Then they had their inspiration. Let's give them a big ovation for proposing DNA was built just like a spiral staircase. They proposed that all our genes are built just like a spiral staircase. Built just like a spiral staircase. Each step made up of a double base. Adenine, thiamine, cytosine, guanine, O. It's called a double helix, Watson said to Crick.
Well, the discovery of the double helix, by any measure, is one of the major moments in the history of mankind. And it's been touted in many ways, but it's famous in part not only for the brilliance of the science of Watson and Crick, but for the beauty of the original paper in 1953 in Nature.
This was a short description of their proposed structure, along with a simple diagram, which they published quickly in order to stake their claim. Towards the end of it, in a now famous understatement, they wrote that it had not escaped their notice that the pairing of complementary bases immediately suggests a possible copying mechanism for the genetic material. In other words, their proposed structure revealed the long-sought secret of heredity. It was easy to imagine that the double helix could split apart, and that each strand could then produce a mirror image of itself through the pairing of A with T and C with G. The end result would be two exact copies of the original double helix.
Told us how the sequence of bases on a chain could be copied. And that had always been the big problem.
They had a foresight about how to translate structure into function, which might have seemed really somewhat foolhardy, but almost every element of which has turned out to be true.
In 1962, Nobel Prizes were awarded to Watson, Crick, and Wilkins. Notably missing was Rosalind Franklin. The Nobel Prize is only awarded to the living, and sadly, she died of cancer in 1958 at age 37. Years later, Sir Aaron Klug, her last and closest colleague, discovered that given a bit more time, Rosalind Franklin might have won the race she didn't know she was in.
See, she didn't publish many papers on DNA, but I have her notebooks here, copies of her notebooks, and I went through them and I saw how pretty close she had got to the answer herself. This is February 1953, and here she's analyzing the distances on the X-ray diffraction photograph, and as you can see, she's trying to decide between a 3-chain or a 2-chain helix, and then concludes that the 3-chain helix is highly improbable. And I have absolutely no doubt that if Watson and Crick hadn't intervened, and I think Crick agrees with me, she would have got it out in due course.
The answer, she would have got the answer over a couple of months if she'd continued to think about it, but she wasn't obsessed by DNA.
We're learning how to pull upon the strings to rewrite the script from which life itself springs. Cracking the code, genetic futures will be foretold. Cracking the code, genetic power about to explode. Cracking the code, genetic mysteries to unfold. Cracking the code, genetic secrets will be told. Cracking the code.